Hello, my name is Sonny Kusaka. I do research in Professor Marina Guenza's lab here at the University of Oregon, and I am going to talk about my project, which is identifying areas of enhanced flexibility in the SARS-CoV-2 spike protein using computational methods. Before I begin, I would like to thank the Undergraduate Research Symposium for giving me the opportunity to present my research here. I would also like to thank the Department of Chemistry and Biochemistry for awarding me with the PChem Undergraduate Research Fellowship for funding over the summer to complete this project. And I would also like to mention that this work has been supported by the use of an extreme science and engineering discovery environment, which is XSEED, which is supported by the National Science Foundation, or NSF. So to begin, I'm pretty sure that we're all very familiar with this virus right here. It is the SARS-CoV-2 virus responsible for the COVID-19 pandemic. And we're interested in studying it for many different reasons, obviously in the development of vaccines and everything, but also it's worth noting this is the third coronavirus that has reached epidemic or worse levels in the past 20 years. So the odds of there being another potential virus that comes along is very high. So the more information we can study about it now, the more we can learn about it now, the better equipped we're going to be to handle another virus like this in the future and potentially prevent a future pandemic. So what I'm interested in is the spike protein, also called the S protein. So we've seen this graphic from the CDC, which has all these little red spikes on them. Well, those are actually the spike protein. And if we zoom in on one of them, it looks like this. It is a very big protein. It's got three big monomers that make this protein a trimer. And I show two images of it here because in order to be infectious to us, the protein structurally rearranges itself. And as we can see, one of the three parts here is actually sticking up here. This is necessary in order to bind to the human cell. And it does that at a receptor called ACE2, or human angiotensin converting enzyme 2. So this is that same open version of that spike protein I showed you earlier. And it has this open and it is connecting to this blue protein right here, which is the human ACE2 receptor. And those are receptors very commonly found, most notably in the oral and nasal mucous membranes, which is why SARS-CoV-2 is an upper respiratory virus. So it binds to human ACE2 via this mechanism right here of structurally rearranging itself and opening it up. I'm curious in studying this because in order to rearrange itself, this has to be some sort of energetically favorable process that's going on. And it would be nice if we could study it on a very small scale where I can zoom in and slow down and see exactly what's going on structurally within the protein for it to do this. So one way by which we can study this is through computational simulations. And basically what they do, they take every atom in the system. So you can take the spike protein, put it in a imaginary box and simulate physiological conditions by adding water and salt, just like you would find inside the human body. And you can solve Newton's equation of motion for every atom in the system at every specific time scale. So like every single nanosecond, you can solve Newton's equation of motion. And this gives a very accurate depiction of how the protein moves in its system over time. And there are a lot of benefits to doing a computational simulation. First of all, it is very safe. There's no actual virus involved. It's the computer simulated virus. So there's zero risk of infection or it getting out. Time can be sort of slowed down in a computer simulation. And what I mean by this is proteins typically perform their functions very quickly by human standards. We're talking within a couple nanoseconds. And this can be very difficult to model. So by using a computer simulation, we can pause whenever we want. We can go frame by frame, like nanosecond by nanosecond, and see what exactly is going on within the protein. Furthermore, we can zoom in. These viruses are very small. The typical SARS-CoV-2 virus is less than 100 nanometers in diameter, I believe. So being able to zoom in and get a really good view up close without the use of super expensive microscopes would be very advantageous for us as well. So other research groups have done computational simulations of the spike protein. One such research group published a paper by Schumark and Colenso et al. And this is a computer simulation using the data that they, so this is from their paper. And basically, this is that atomistic simulation where every atom, Newton's equation of motion, has been solved over nanoseconds. And this is a 200 nanosecond simulation. So the protein just sort of wiggles. And you can see some movement here. And this is what it's doing on the SARS-CoV-2 virus. The downside with computer simulations is that they can be tough to analyze. So for example, we see this protein wiggling and moving, and we can see how it moves and whatnot. And that's very good. That's good information. But which of these movements are important? Which of these movements are really important for it to bind? And that can be very tough to look at just based on this wiggling I see, because I can't really say anything else other than the protein wiggles around a bit. So analysis techniques have been developed that can analyze the trajectory of the computer simulation and thereby give us a better sort of idea of what's going on within the protein. So the methods that I used all fall under the theory that flexibility more or less equates to functionality. And this was a hypothesis by Monod, Wyman, and Chanju. And basically what this says is that they developed a theory in which the more flexible a region of the protein is, the more likely it is to exhibit binding characteristics. High flexibility means that the part of the protein that's flexible can occupy many different sort of conformations in space. So the more conformations in space it can occupy, the more likely it is to be able to bind to something. So I used 3 different methods. One is the Gaussian network model, principal component analysis, and then also the Langevin equation for protein dynamics. The math that goes into these models is a bit beyond the scope of this presentation, but the main takeaways here are that the Gaussian network model is expected to be the least accurate but easiest to perform. PCA is kind of in the middle of the two, and the LE4PD should be the most accurate. However, it is also the most difficult to perform. So you might have the question of why then would I want a analysis? Why would I bother with the GNM and PCA if the LE4PD is supposed to be the most accurate? Well, the LE4PD is actually a pretty new technique. It was developed here at the University of Oregon in the Guenza lab by Copperman and Guenza. And it has been successfully implicated in measuring the dynamics accurately of small proteins like HIV protease, for example. That's a very small protein. It's about 100 amino acids in length. The spike protein is much bigger than that. It has over 3,300 amino acids. And just because a model works for a small protein doesn't necessarily mean it's going to work for a large protein as well. So that was one of the goals of this research, was to be able to see if this would be a good model to model a really large multi-meric sub protein like the spike protein. So the research has been published that has identified the functional domains of the protein. There are these regions right here. Their functions are not super important for this presentation, but these regions have been color-coded based on what areas have a defined biological function. What is most notable here is this sort of top half, the red, the orange, and the cyan. Those are the regions that actually interact with the ACE2 human receptor. So based on the flexibility equals functionality theory, these should be the most flexible regions of the protein as predicted by all 3 of the models. And when we look at simulations of the closed conformation, here's the Gaussian network model, here's PCA, here's LE4PD. Warmer colors mean that higher flexibility. And the Gaussian network model is, it shows highly flexible regions right here in the N-terminal domains. PCA, we can see it's actually a bit more detailed. So it doesn't just say, oh, this entire region should be flexible. We can actually see sort of on a more localized basis where the flexibility lies. And with LE4PD, it's a bit hard to see here, but it's even more detailed than PCA. And that's exactly what we would expect going into this. This should be the most accurate, and so far it looks like it is. Here's the open conformation. This is the conformation that actually binds to the human ACE2 receptor. And as we can see, the GNM, this shows a very, very flexible monomer right here, and this is the one that binds to the human receptor. PCA, we can see it doesn't predict quite the same thing, but it predicts all of the same general areas as the GNM. And the LE4PD, once again, is the most detailed out of the three. It doesn't just say this entire region is flexible like GNM and PCA. It actually goes residue by residue basis and says this should be flexible, this part's not, this part is, this part's not. So we get a much better idea of what's happening within the protein. And probably one of the most interesting things is that most of this flexibility across the board is seen in the regions with a characterized functional domain. So the parts that are actually biologically most important for the protein to perform its function. However, the Langevin equation for protein dynamics did identify several regions outside of the functional domains that were highly flexible. And I've labeled them right here. These AA stands for amino acids, and it's just this sequence right here that shows high levels of flexibility. These are all generally loop structures, and they're on the exterior surface of the protein. So it's not surprising that these are flexible. But whether or not these are important for the functionality of the protein, I don't know. These regions have not been previously implicated as having a function for the protein in the literature. So it would be very interesting if these regions were important for the protein to do its biological function. And if so, they could potentially be regions for drug targeting, for example, which is when you take an inhibitor drug and bind it to a part of the protein, and then you can sort of make it rigid, stop it from moving, and thereby stop it from performing its biological function. So the main takeaways, the LE4PD does appear to be a promising research technique for analyzing very large multi-subunit proteins like the spike protein, which is very good. This could potentially open up a lot of doors in the future for using it as a research technique. Also, the LE4PD analysis identified regions of enhanced flexibility that fall outside of the previously characterized functional domains. So whether these are actually super important for the protein or not, we're not sure, but this has definitely narrowed down based on certain criteria of the criteria of flexibility. This has narrowed down our view of, well, what is functional in this protein, and are we able to now take this data and learn from it in the future? So these are the works that I referenced in performing this research. Thank you very much for watching.